Every year, the supplement industry sells billions of dollars worth of anti-aging products, powders, capsules, serums, most of them built on compounds your body barely absorbs, backed by studies conducted in petri dishes, and priced like they came down from a mountaintop. And every year, people buy them because the promise feels urgent. Because aging feels urgent, here's what almost nobody in that industry wants you to notice. In 2016, a Japanese scientist named Yoshinori Ohsumi won the Nobel Prize in Physiology, or Medicine, for discovering the genetic machinery behind a process that your body already knows how to do, a process that clears out damaged, aging cellular components, essentially the body's own renovation crew. And the compound that most consistently switches it on, it's not patented. It's not sold at a premium. It has been sitting in legumes, mushrooms, fermented foods, and whole grains for as long as human beings have been eating. Today we're breaking down 13 of the most important foods linked to this Nobel Prize discovery, ranked by actual peer-reviewed food chemistry data, not blog estimates. Pay close attention to number 1 on this list. Most people have had it in their kitchen their entire lives and never once thought of it in this context. And when we reach the middle of this list, I'm going to tell you the story of a food that Ming Dynasty physicians in China were prescribing specifically for the fatigue of aging 600 years before anyone had ever heard the word autophagy. Stay with me. This one goes deep. These are 13 foods where the science is real. The history runs deep, and the preparation matters more than most people realize. Number 13. Popcorn. This might be the entry that makes you stop and reread the title, but it belongs here, and here's why it matters that it does. Popcorn, specifically air-popped corn, appears in the peer-reviewed food chemistry databases that researchers have used to rank spermidine content across real foods. The amount per serving is modest. This is not the food that anchors a longevity strategy, but the fact that it's here at all tells you something important about the compound itself. Spermidine is not rare. It's not exotic. It is, in the most literal sense, an ordinary presence in ordinary foods, and that's precisely the point. Corn itself has been a sacred food across Mesoamerican cultures for thousands of years. The Popol Vuh, the creation text of the Quiche Maya, describes human beings as literally made from maize, not metaphorically. But cosmologically, the Aztecs offered maize at the festival of Huitzilopochtli, understanding it as a sustaining gift between the divine and the human. They weren't describing cellular biology. They were describing dependency. A civilization that could not survive without this food understood it to be foundational in a way that modern nutritional science is only now beginning to quantify. The practical note here is significant preparation strips the benefit or preserves it. Air-popped, lightly salted. Unbuttered popcorn carries real spermidine. The version soaked in processed oils and artificial flavoring is a different food entirely. The kernel is the same. The chemistry is not. 1 tablespoon of nutritional yeast itself, a modest spermidine source, sprinkled over plain popcorn, adds a savory, almost cheese-like flavor, while layering in B vitamins that support the cellular energy system's autophagy depends on what the Maya understood intuitively and what the food chemistry data confirms quietly. Ordinary foods carry extraordinary functions. The question is always what you do with them. Number 12. Pear. Pear has been a healing food in traditional Chinese medicine for over 2,000 years, and its use was remarkably specific. Classical texts from the Tang Dynasty describe pear decoctions prepared to calm what practitioners called lung heat, the dryness, inflammation, and respiratory vulnerability that comes with illness or aging. It was prepared as a slow-simmered broth, with the skin on, sometimes with ginger and honey, to treat what we might now describe as upper respiratory inflammation. What strikes researchers who study the convergence of traditional food systems is that this was not an isolated practice. Medieval Persian physicians working within the tradition of Ibn Sina, whose medical encyclopedia remained a standard reference in European universities until the 17th century, also used pear therapeutically, specifically for digestive clearing and what they call dampening internal fire. Two medical traditions, separated by culture and geography, both reaching for the same fruit for overlapping reasons. Modern food chemistry analysis confirms that pear carries measurable spermidine, alongside significant fiber and vitamin C. What the research also shows, and what most wellness content skips, is that the skin is not optional. The outer peel of a pear concentrates both fiber and polyphenols at significantly higher levels than the flesh alone. When you peel a pear before eating it, you are discarding a meaningful portion of what makes it nutritionally distinct. Eat it whole. 1 pear. Skin on. Eaten in the morning before food. Allows the fiber to act as a prebiotic primer for the gut bacteria that independently produce small amounts of spermidine as a fermentation byproduct, effectively giving you two sources from one piece of fruit. The Tang Dynasty physician didn't need a food chemistry database to know this fruit mattered. Careful observation across generations was its own form of clinical trial. Number 11. Whole grain bread. Among the most ancient foods in human civilization, bread carries a symbolic weight in Scripture that is matched by very few other foods. In the Gospel of John, Jesus describes himself as the bread of life. In Exodus, manna, the bread that fell from heaven in the wilderness, sustained an entire people for 40 years. In Leviticus, the showbread was placed before God, each Sabbath as an offering. Bread, in the Hebrew and Christian traditions, was not incidental nourishment. It was the primary image of divine provision itself. What matters for our purposes is what kind of bread those ancient peoples were eating, not refined white flour, which didn't become widely available until industrialized milling in the 19th century. They were eating whole grain bread, dense, dark, bran-intact loaves that retained the full nutritional architecture of the wheat kernel. And it is that whole grain architecture, specifically the germ and bran fractions, that carries the spermidine content. Multiple European food analysis studies, including research from the United Kingdom, Norway, and France, have measured whole grain bread's spermidine content. The numbers show moderate but consistent levels across different studies and different bread types, which gives this entry more statistical confidence than single study entries on this list. The consistency across multiple research groups in multiple countries is the thing to pay attention to. It suggests this isn't an artifact of one lab's methodology. Practical specificity. Choose bread, where the first ingredient is a whole grain flour, where you can see the grain texture in the crumb, and where the ingredient list is short enough to read aloud in one breath. A slice with a thin spread of raw tahini itself, a modest spermidine source, adds a second layer from the same meal with a flavor combination used in Middle Eastern cooking for centuries. Number 10. Cauliflower. Cauliflower belongs to the Brassica family, a genus of vegetables with one of the longest cultivation histories in the Mediterranean world. Roman agricultural writers, including Pliny the Elder, described Brassica vegetables as among the most important foods in their diet. In Ayurvedic medicine, vegetables from this family were described as cooling foods used to reduce excess pitta, the metabolic fire that, when unbalanced, was believed to drive inflammation and early aging. The spermidine content in cauliflower is moderate but consistent across multiple European food analysis studies. What makes this entry particularly interesting to researchers isn't the spermidine alone, it's the convergence of active compounds in a single food. Cauliflower also contains glucosinolate, the sulfur compounds that convert in the body to isothiocyanates. These are the molecules that activate NRF2 pathways, your cell's own master antioxidant switch, through a completely separate mechanism from spermidine's autophagy trigger. You are, in effect, running two parallel cellular maintenance protocols from one vegetable. There is a preparation detail here that almost no one discusses. Chopping or crushing cauliflower and then waiting 10 minutes before cooking allows an enzyme called myrosinase to convert glucosinolates to their active isothiocyanate form. If you put cauliflower directly from the cutting board into a hot pan, that enzyme is deactivated before conversion occurs, and you lose a meaningful portion of one of the compounds that makes this vegetable worth eating. Chop, wait, then cook. 10 minutes of patience changes the chemistry of the meal. The ancient physician, observing that brassica vegetables seemed to support vitality across generations, wasn't making a guess. They were, without knowing it, identifying a plant that runs multiple biological programs simultaneously. Number 9. Red kidney beans. Beans have fed humanity for longer than almost any other cultivated food. In Mesoamerica, the common bean ancestor of kidney beans was part of the three sisters, maize, squash, and beans, planted together, sustained together, eaten together. Indigenous agricultural traditions understood intuitively that these three foods completed each other. What we now know is that the lysine in beans offsets the amino acid gaps in corn, creating a complete protein profile that sustained entire civilizations without animal protein. They didn't call it protein complementarity. They called it the gift of the sisters. In Mediterranean cooking, white and red beans have been a dietary staple since ancient Greece. Hippocrates, the same physician who gave medicine its foundational ethical oath, wrote about the importance of legumes in the daily diet, describing them as foods that build the body steadily over time rather than quickly, which he believed to be a mark of their deeper value. UK food chemistry analysis confirms kidney beans among the more concentrated legume sources of spermidine. Alongside resistant starch and anthocyanins, the red pigments in the seed coat that carry independent antioxidant research. The color matters. The red coat that makes kidney beans visually distinctive from white beans is precisely where a significant portion of their polyphenol activity lives. When you make a bean dish and drain the cooking liquid, you're pouring a meaningful fraction of those water-soluble anthocyanins down the drain. If you cook from dried beans in their own liquid, or use that cooking liquid as a base for soup, you retain far more of what makes the red coat worth eating. Chili with kidney beans, slowly simmered in their own broth, with the liquid incorporated rather than discarded. That's not just a recipe. That's following the logic of the plant. Number 8. Mushroom. This is the one I promised you. Mushroom cultivation for medicinal purpose in China traces back roughly 1,000 years. But it was during the Ming Dynasty, between 1368 and 1644, that shiitake mushrooms specifically became a widely adopted tonic food prescribed by physicians to counteract the aches and fatigue that were understood to come with aging, and credited with supporting heart function and lung capacity. The Ming Dynasty medical text, Bensao Gangmu, compiled by physician Li Shizhen over 27 years and published in 1596, included detailed descriptions of mushroom use for reinvigorating the body's vital energy, what practitioners called qi, in elderly patients. Independently, on the other side of the world from that tradition separated by ocean, language, and culture, mushrooms were documented as a medicinal gift to the Japanese imperial court as early as the year 199 CE. By the Edo period, mushroom cultivation had become integrated into Kampo, Japan's traditional medicine system, where specific mushroom species were prescribed with a precision that rivaled pharmaceutical thinking. Two civilizations with no documented contact in this period, both reaching for mushrooms specifically as an aging, support food, both attributing the same categories of benefit: vitality, heart support, immune resilience to the same class of organism. The chemistry begins to explain why. Japanese food analysis data shows mushrooms carrying one of the higher spermidine concentrations per 100 grams of any measured food category. Beyond spermidine, mushrooms bring beta-glucans, long-chain polysaccharides studied extensively for their effects on immune regulation, specifically their interaction with macrophages and natural killer cells. And then there is ergothioneine, an antioxidant amino acid that mushrooms are the primary dietary source of, which has its own growing body of aging research, suggesting it may accumulate specifically in tissues under high oxidative stress: the brain, the liver, the kidneys. Ergothioneine is sometimes called a longevity vitamin by researchers studying it, though that designation remains preliminary. Preparation matters here in a way that surprises most people. Drying mushrooms and then reconstituting them in warm water concentrates the beta-glucan content dramatically compared to fresh. The soaking liquid itself, when strained and used as a broth base, carries significant amounts of both water-soluble compounds and the savory glutamate compounds that make mushroom broth one of the deepest flavors in any kitchen tradition. Discard the soaking liquid, and you discard part of what makes dried mushrooms worth using. The Ming Dynasty physician working by oil lamp in a mountainside monastery did not have mass spectrometry. He had patients across decades of careful observation. The research is only now beginning to understand what he found. If this kind of connection between ancient observation and modern science is the kind of thing that makes you lean forward, hit like and subscribe right now. Because what's coming in the next few entries goes even deeper into the evidence, and I'm going to be honest with you about exactly where it's strong and exactly where it still has work to do. Number 7. Lentils. Among the oldest cultivated legumes in human history, lentils appear in Genesis 25 inches, one of the most vivid and psychologically dense scenes in all of Scripture. Esau, the elder twin, returns from the field exhausted and famished. The Hebrew word used is ayef, a word that implies bone-level depletion, not simple hunger, and asks his brother Jacob for that red stuff. Jacob, reading the desperation in his brother's face, offers a bowl of red lentil stew in exchange for Esau's birthright. Esau agrees. Theologians have argued for millennia about what this exchange means about Esau's character, about birthright, about impulsivity and wisdom. But set aside the theology for a moment and look at what the text is actually describing. A man at the edge of physical collapse is restored by a bowl of lentils. That isn't just narrative color. That's a description of what a protein-dense, iron-rich, folate-containing, slowly digesting legume actually does in a depleted human body. The ancient world knew this food's power viscerally, through repeated experience. The spermidin content in lentil soup, measured in published food chemistry studies, places it solidly in the upper half of the ranked food list. And lentils bring something else. The science is increasingly interested in resistant starch, which ferments in the large intestine and feeds the specific bacterial species, Bifidobacterium and Lactobacillus, that independently produce short-chain fatty acids, including butyrate, a compound with its own studied role in cellular inflammation regulation. Red lentils, which cook in under 20 minutes without soaking, need no more preparation than this: simmer 1 cup in 2 cups of water, with a pinch of turmeric and a 1/4 teaspoon of black pepper. The black pepper, specifically its piperine, increases curcumin absorption from turmeric by up to 2,000%, according to pharmacokinetic studies. You are, in effect, combining the spermidine of the lentils with the anti-inflammatory research profile of curcumin in a bowl that takes 20 minutes and costs almost nothing. Esau could have asked for many things. He asked for lentils. Perhaps hunger is its own form of ancient wisdom. Number 6. Natto. Here is a food with one of the richest documented histories on this entire list, and also one of the most significant concentration of active compounds per serving of anything we'll discuss today. Natto is fermented soybeans, and its origin story carries 2 competing traditions. The first holds that it arrived in Japan from China during the Nara period in the 700s CE, introduced through Buddhist monastic exchange networks. The second traces it to a specific moment in 1083 CE, when a samurai general named Minamoto no Yoshii, on campaign in northern Japan, had his men boil soybeans for the horses. The story goes that an unexpected warm spell triggered fermentation overnight in the straw bags used for storage. The soldiers ate the result, not knowing what had happened, and found it to their liking. What is historically certain by the Edo period 1603 to 1868 natto was sold fresh by street vendors in Japanese cities every morning. It was ordinary breakfast food, and it became an integral part of the devotional diet of Zen Buddhist monks who maintained strict vegetarian practice and needed concentrated, complete protein from plant sources. The spermidine content of natto is consistently reported as among the highest of any measured food driven specifically by the fermentation process, in which Bacillus subtilis var. Natto bacteria break down proteins and release polyamines as a metabolic byproduct. Fermentation is, in a very real sense, a spermidine production process happening in the food itself before it ever reaches your digestive system. But natto carries a second compound that has its own entirely independent research basis, vitamin K2, specifically the MK7 form, which is found in almost no other common food in significant amounts. MK7 has been studied for its role in directing calcium into bones rather than arterial walls, a distinction that carries genuine clinical interest in cardiovascular and bone health research. Natto also contains nattokinase, an enzyme studied for its effects on fibrin, the protein involved in blood clot formation, with results that researchers describe as worthy of continued investigation. One serving of natto, approximately 100 grams over warm rice, with a small amount of tamari and a touch of prepared mustard, which enhances the flavor profile dramatically. Eaten in the morning, is doing multiple biological jobs simultaneously. Its strong flavor and sticky texture are polarizing. It is also, by the data, one of the most nutritionally dense fermented foods that exists. Number 5. Green peas. Peas have fed Eastern Mediterranean and Near Eastern populations for over 10,000 years, placing them among the earliest cultivated plants in human agricultural history. Archaeological evidence from sites in the Fertile Crescent suggests pea cultivation predates written language. They appear in Bronze Age food stores, in Roman agricultural manuals, and in the diets of Mesopotamian societies that saw them as foundational, sustaining food rather than a side dish. What modern peer-reviewed food chemistry shows is that green peas, fresh or frozen, rank as one of the highest spermidine content foods measured per actual consumed serving. In the data compiled by research groups studying dietary spermidine intake, peas consistently appear near the very top of the ranking, behind only soybeans in some analyses. This is not a compound present in trace amounts. It is a food where the concentration is genuinely meaningful. There is a preparation detail almost no one mentions. Frozen peas, which are blanched and frozen within hours of harvest, test consistently for spermidine retention. Fresh peas, if they've been sitting in warm storage for several days, may have degraded more significantly. The blanching that people often assume diminishes fresh vegetables turns out, for peas specifically, to preserve more of the heat-stable compounds than slow ambient degradation does. Buy frozen with confidence. The convenience and the chemistry are aligned. Add peas directly into warm soups in the final 2 minutes of cooking, not at the beginning, to preserve both color and compound integrity. Or eat them cold, straight from the bag after thawing, with a squeeze of lemon and a drizzle of cold-pressed olive oil. The Mediterranean didn't need a food science laboratory to figure out that this combination worked. Number 4. Aged cheese, specifically Parmesan. Parmesan-style cheese production traces its documented history to the Middle Ages. In the Emilia-Romagna region of Italy, where Benedictine and Cistercian monks developed a long-lasting hard cheese using monastery milk and local sea salt. Production is documented in written sources from approximately the year 1200 CE. Before that, Roman legionaries were already carrying hard salted aged cheese on military campaigns, not because they understood its biochemistry, but because it was dense in calories, resistant to spoilage, and lightweight enough to carry across hundreds of miles. The chemistry of aging in cheese is genuinely interesting and rarely explained. As cheese ages, bacteria break down proteins through proteolysis and release polyamines, including spermidine, as a metabolic byproduct. A fresh mozzarella tests near zero for spermidine. A 2-year aged Parmigiano Reggiano tests dramatically higher. The aging process is, literally, a spermidine production process. Time is an ingredient. The published food chemistry literature is honest about something important. The numbers vary enormously between studies, between cheeses, between countries. And between aging times, some analyses report figures many times higher than others. What remains consistent across that variation is the direction of the finding. A large American population study, tracking over 20,000 adults, specifically found that spermidine coming from cheese alongside vegetables, legumes, and nuts was associated with lower cardiovascular and all-cause mortality over the study period. The researchers described the association as worthy of further prospective investigation. 20 grams of genuinely aged Parmigiano Reggiano, not the pre-graded shelf-stable version, which has been processed in ways that alter its compound profile graded over a bowl of legumes or whole grain pasta, is doing biochemical work while being, simultaneously, one of the most pleasurable flavors in the world. The monks who developed this cheese were not thinking about polyamines. They were thinking about feeding a monastery through winter. They solved both problems without knowing they were related. Number 3. Wheat germ. If you've encountered the word spermidine before today, there's a reasonable chance it was attached to this food. Wheat germ is the most concentrated common source of spermidine that researchers have formally measured, and it is also the form used in the one rigorous human clinical trial that has directly tested whether dietary spermidine intake influences cognitive aging. That trial, run out of the Neurological Clinical Research Center at Charité in Berlin, gave older adults with mild cognitive decline a standardized wheat germ extract, supplying under 1 milligram of spermidine per day for 12 months. The results, and I'm telling you this because a channel that only shares the studies that agree with the title isn't giving you science. It's giving you marketing with citations, did not show a statistically significant benefit over placebo on the primary cognitive endpoint measured. The trend was in the right direction. The effect size was not strong enough to meet the bar for significance. The researchers themselves described the result as preliminary and called for longer trials. I am including this information not to undermine the other 12 foods on this list, but because this is the kind of honest accounting that actually builds trust. The broader population data, 2 separate long-term studies, one tracking over 800 people in Brunette, Italy for 2 decades, and another following nearly 24,000 American adults, both found that higher dietary spermidine intake was associated with meaningfully lower rates of death over the study period. That is genuinely encouraging signal. The clinical trial is the next level of evidence, and that evidence is still developing. Wheat germ, stripped out of refined flour during industrial milling, is the small nutrient-dense core of the wheat kernel. A tablespoon stirred into oatmeal or yogurt has a mild, slightly nutty flavor you'll barely notice. It carries, alongside spermidine, vitamin E in its most bioavailable tocotrienol forms, zinc, magnesium, and folate, all nutrients with independent research bases that have nothing to do with autophagy. The Hebrew scriptures describe wheat as one of the 7 species of the land of Israel, foods of special significance in both ecological and spiritual terms. The first fruits offering of wheat was brought to the temple. The wheat kernel was understood to contain the life force of the grain. Modern milling removes the germ to extend shelf life. The ancient world ate it whole, without knowing why that mattered. The research is only now beginning to quantify the difference. Number 2. Soybeans. When researchers built the most comprehensive, peer-reviewed database mapping spermidine content across real-world portion sizes, not laboratory concentrations, but what a human being actually eats in a single sitting, cooked soybeans came out at the top of every food category measured. By a meaningful margin, soy has been cultivated in China for at least 5,000 years. The Shenan Benchao Jing, one of the foundational texts of Chinese medicine, lists soy among what it calls the 5 sacred grains, along with rice, wheat, barley, and millet, describing each as essential to human vitality and longevity. The text was compiled in the early centuries CE, but is attributed to the legendary Emperor Shennong. Believed to have introduced agriculture to humanity, soybeans fed the East Asian population for millennia. They moved through trade routes to Korea, Japan, and eventually Southeast Asia. where their fermented forms miso, tempeh, natto became distinct culinary and nutritional traditions. What the food chemistry data confirms is that fermented soy products amplify the spermidine story significantly. Fermentation by specific bacterial strains does what aging does for cheese. It liberates bound polyamines and dramatically increases the concentration available per serving. Natto, which we discussed earlier, is fermented soy. Miso is fermented soy. Tempeh is fermented soy. This family of foods represents an entire ecosystem of spermidine delivery systems, and every one of them has a history of use spanning centuries to millennia. The large American population study I mentioned, tracking nearly 24,000 adults over time, specifically named legumes, including soy, as one of the food categories whose dietary spermidine intake correlated with lower cardiovascular and all-cause mortality. Edamame as a snack. Whole cooked soybeans in a grain bowl. Tempeh, sautéed with ginger and tamari. Miso stirred into warm, not boiling water with a piece of kombu. The preparation options are as varied as the civilizations that have relied on this plant after 19 other foods, many of them surprising. The single highest spermidine source per real portion is one of the most ordinary legumes on Earth. That is not anticlimactic. That is the point. Number 1. Wheat germ plus soybeans plus natto, the triad that started this research. I want to use the final entry on this list differently, not as a new food, but as a lens through which to see everything we've covered today. The research group in Graz that has most consistently investigated dietary spermidine and longevity outcomes keeps returning to a specific pattern in their population data. The people with the highest dietary spermidine intake are not eating exotic superfoods. They are not spending money on premium supplements. They are eating more legumes, more whole grains, more fermented foods, more aged cheeses in small amounts, more mushrooms, more vegetables from the brassica family. This is a dietary pattern that has existed in various forms across cultures for thousands of years. The Mediterranean diet, traditional Japanese dietary patterns, the diet of the Fertile Crescent, they converge on these same food categories. Not because any ancient physician knew about autophagy, because careful observation across generations identified these foods as the ones that seemed to track with longer, more vital life. What the Nobel Prize-winning science has given us is not a new discovery. Exactly. It is a mechanism, a biological explanation for something that observant human beings have been noticing and recording for millennia. Ohsumi found the machinery. The foods that trigger that machinery were sitting on tables in Tokyo, Athens, Jerusalem, and Beijing, long before anyone could explain why they worked. Here is the one thing I want you to carry with you from this video, and it matters more than any individual food on this list. Processing degrades spermidine. Heat denatures it. Refinement removes it. The wheat germ that makes whole grain bread a meaningful spermidine source is the exact fraction removed when wheat is refined into white flour. The aging that makes hard cheese a concentrated source is bypassed entirely by fresh, processed cheese product. The fermentation that elevates natto and miso is stripped away when soy is processed into soy protein isolate. In almost every case on this list, the traditional preparation, whole grain, fermented, aged, minimally cooked, preserves what the modern industrial version destroys. This is not a coincidence. It is a pattern, and it runs through almost every conversation about food and aging that is worth having. The ancient world ate these foods in their most intact, most fermented, most whole forms, not because they understood polyamine chemistry. They ate them that way because that was the only way they existed. Modern processing invented the removal, and in removing what seemed inconvenient, the bran, the germ, the fermentation time, the aging period, it removed, without knowing, a significant portion of what made the food biologically active. The question worth sitting with after this video, the one I'd like you to take into your next trip to the grocery store or your next meal is this. How many foods in your daily life are the refined version of something that once carried a compound the science is only now beginning to understand. You don't need to overhaul everything at once. You need to make one or two choices, consistently, that move in the direction of whole, fermented, aged, and minimally processed. That's it. That's the strategy. If this video gave you something you can actually use, not just something that sounds impressive, but something you'll think about tomorrow, please hit that like button. Share this with someone in your life who is paying attention to their health right now. Drop a comment telling me which food on this list surprised you the most. Are you Team edamame, Team natto, Team mushrooms? Or did the wheat germ genuinely catch you off guard? And if you haven't subscribed yet, click that button and ring the bell. Because we'll keep doing exactly this, connecting the science that's real to the foods your body was built to use, and being honest with you about exactly where the evidence is strong, and exactly where it still has work to do. The compound that helped win a Nobel Prize is not locked in a laboratory or a supplement bottle. It has been sitting on dinner tables, in monastery kitchens, in samurai provisions, and in the stew that a depleted man traded his birthright for in a field outside Canaan. Take care of yourself from the inside.